This video covers Chapter 11, which is cell communication. We will look at cell communication, signal transduction, and changes in signal transduction pathways. What we're looking at here is how do cells communicate, and we'll start by looking at communications among microorganisms, specifically yeast. Yeast identify their sexual mates by chemical signaling. There are two mating types, A and alpha. Each type secretes a specific factor that only binds to the other cell type. When exposed, the opposite type mating pairs change shape, grow towards each other, and fuse. And the new A/alpha cell contains all the genes of both the original cells. The match between mating factor and receptor is key to making sure mating occurs among cells of the same species of yeast. And this process occurs in a series of steps called a signal transduction pathway. This is super similar to the pathway found in animals, even if the purpose is different, which is more evidence that we are all from the same common ancestor. It likely evolved in ancient prokaryotes and single-celled eukaryotes. For more information on prokaryotic cell signaling, please watch the video on your Unit 4 guide on quorum sensing. It is super cool, and it is about how bacteria communicate with each other. So this is a picture of the yeast mating communication. We have our A cell and our alpha cell. You can see that the factor they're secreting is different in each cell type, and that the factor will fit into the receptor on the opposite cell type. So they will secrete their factor, they bind, and then we have our new A alpha cell at the end. Multicellular organisms communicate using signaling molecules. These will be targeted for specific cells, and they can either be close to or far from the signaling cell. With local signaling, cells are in direct contact. So we're looking at cell junction where the signaling molecules pass readily between the two cells, or cell-cell recognition where they communicate by molecules on their surface. Cell-cell recognition is important for embryonic development and our immune system's responses. So we can see in this picture the cell junction, the cells are literally connected by a junction, and so those molecules can just go back and forth as they please. With cell-to-cell recognition, one cell has something that will fit into something on the other cell, so they can communicate that way. Most often, signaling molecules are secreted by a signaling cell. There are two types of local signaling that we'll look at: paracrine, which happens in animal cells, where local regulators influence cells in the vicinity. An example of that would be growth factors will stimulate nearby cells to grow and divide. And synaptic signaling, which also happens in animal cells. During synaptic signaling, electrical signal triggers secretion of neurotransmitters that triggers a response in the target cell. So that's how our immune system works. An example of long-distance signaling is endocrine signaling, which happens in both plants and animals, where specialized cells release hormones that travel to other parts of the body that will recognize and respond to the hormone. With plants, those hormones will travel around via vessels, and obviously with animals, it's going to travel through the blood. And since not every cell will have a receptor for whatever that hormone is, that hormone will only cause a response in cells that have a receptor for that specific hormone. Here are picture examples of all three of those things: paracrine, synaptic, and endocrine signaling. As a preview, because we're getting into a little bit of complicated things, there are 3 steps to cell signaling. The first part is called reception, where the target cell detects the signaling molecule from outside the cell. Then we have transduction, where the binding of the signaling molecule changes the receptor protein, and the signal is converted into a form that can bring a cellular response, which is usually a series of steps, as we will see. And then the response, where the signal triggers a specific cellular response. Even though the exact thing that is happening, depending on what the signal and the response are, is very, very different, the general pathway of cell communication is quite similar between signals and organisms. This is our overview picture. We have something binding to a protein, this middle bit where the signal is relayed, and then finally the response. So let's start with reception. Signaling molecules act as a ligand. A ligand is any molecule that binds to a larger one. Ligand binding usually causes the receptor protein to change shape, and that shape change can directly activate the receptor or cause a chain reaction. Most receptor proteins are on the cell surface, but some are on the inside of the cell instead. And we'll look at what happens with both of those cases. The largest family of human cell surface receptors are called G protein coupled receptors, or GPCRs. We will also look at two other of those, which would be the receptor tyrosine kinases and ion channel receptors. But the picture here on the right shows a G protein coupled receptor. So you can see the plasma membrane, and then that blue bit is the actual receptor, and the orange bit is cholesterol, which, as we know, is found scattered throughout the plasma membrane. These are all different ways that receptors can, have a ligand bind to them. I do not want to spend the 20 or 30 minutes it would take to cover these right now, so please take a few minutes to review figure 11.8 in your book and be somewhat familiar with these different cases. Intracellular receptor proteins are found in the cytoplasm or nucleus of the target cells. Signaling molecules will enter the target cell's plasma membrane, and these signaling molecules are small and nonpolar enough to just be able to go through the membrane without having to go through a protein. Some examples of these types of signaling molecules are steroid hormones, thyroid hormones, and nitric oxide. After the molecule enters a cell, it binds to an intracellular receptor, meaning it's on the inside, like a hormone receptor complex, which will cause a response. Usually here the response would be turning a gene on or turning a gene off. This is an example of an intracellular receptor for aldosterone. The adrenal gland secretes aldosterone. Aldosterone travels through blood, but it only is going to cause a response in kidney cells because that's where the receptors for aldosterone are. It binds and activates receptor proteins in kidney cells. The active form of the receptor protein enters the nucleus, and it turns on genes that control water and sodium flow in the kidney cells, which ultimately affects blood volume, which you can see in the picture as well. So the hormone just squiggles right on through the plasma membrane, binds to a receptor protein, makes its way into the nucleus, and then turns a gene on. So the DNA is transcribed into mRNA and then translated into a protein. I know we haven't gone over protein synthesis yet, but we will shortly after winter break. Moving on to the second step of cell signaling, transduction. So we're going to be looking at cascades of molecular interactions, relay signals from receptors to target molecules in the cell. Transduction is usually a multi-step pathway that involves a lot of molecules. It often includes activation of proteins by addition or removal of phosphate groups or release of other small molecules or ions that will act as signaling molecules. The benefit of this process is that it's able to amplify the signal, and it does have some regulation on the response. What we don't want to happen is the same signal being sent over and over again if the response is no longer necessary. So we do need to have some control at every bit of this process. As an overview of the signal transduction pathway, signal-activated receptor activates another molecule, which activates another molecule, and this keeps going until the protein that produces the final response is activated. Relay molecules will relay the signal from receptor to response, and these are often proteins, but they do not have to be. And just as an FYI, the original signaling molecule never enters the cell. The ligand binds to the receptor protein, which changes shape, and that shape change is what elicits all of this. So the ligand is never going to enter the cell unless we are talking about what we had talked about previously, where the thing is able to just go through the plasma membrane. But discarding all of that, if this is happening, is not because that thing, the ligand, has come into the cell. Let's look at protein phosphorylation and dephosphorylation. Phosphorylation and dephosphorylation of proteins is a widespread mechanism for regulating protein activity. An enzyme that transfers phosphate groups from ATP to a protein is known as a protein kinase. The receptor tyrosine kinase is a specific kind of protein kinase that phosphorylates tyrosines, which is an amino acid. Most cytoplasmic protein kinases act on proteins that are different from themselves. Most phosphorylate serine or threonine rather than tyrosine. Many relay molecules in a signal transduction pathway are protein kinases that act on other protein kinases in the pathway, which leads to a phosphorylation cascade. The shape changes alters function of the protein, usually activating it, but it also could be a deactivation. So here is a picture example of a phosphorylation cascade. We have our signaling molecule that binds to a receptor, which activates a relay molecule. That relay molecule will activate protein kinase 1. The active protein kinase 1 activates protein kinase 2. The active protein kinase 2 phosphorylates another protein that brings about the cell's response to the signal. So we just have all of those protein kinases being activated and so that they can activate something else, on and on and on, until we have our final thing that leads to a cellular response. And then we can remove those phosphate groups so that those molecules can be reused in another phosphorylation cascade later. Protein phosphatases are also important in the phosphorylation cascade because they will remove the phosphate groups from proteins, meaning dephosphorylation. This inactivates the protein kinases, which make them available to be able to be reused later on. A lot of signaling pathways will require small non-protein water-soluble molecules or ions, and these are called second messengers, where the first messenger would be the ligand. They are readily spread all over the cell via diffusion, and they are part of the pathways initiated by G protein-coupled receptors and receptor tyrosine kinases. The two most used are cyclic AMP or CAMP and calcium ions. Lots of relay proteins are sensitive to changes in concentrations of second messengers in the cytoplasm. So let's look at an example with cyclic AMP. Cyclic AMP stands for cyclic adenosine monophosphate. Adenylyl cyclase is the enzyme in the plasma membrane that converts ATP to CAMP in response to an extracellular signal like epinephrine, which we can see here. So we have ATP, and our enzyme removes two of those phosphate groups to make cyclic AMP, and then phosphodiesterase, which is yet another enzyme, will convert that into AMP, just regular AMP. Here is an example of cyclic AMP as a second messenger in a G protein signaling pathway. So we have our first messenger, which is our signaling molecule. Here is epinephrine, binds to the GPCR, and then which activates it. The activated GPCR binds to G protein, which is then bound by GTP, which activates the G protein. The activated G protein / GTP binds to adenylyl cyclase. GTP is hydrolyzed, which activates the enzyme. The activated enzyme converts ATP into cyclic AMP, And then CAMP, which is now our second messenger, activates another protein, which gives us our cellular response. Looking at calcium ions and IP3 next. Many signaling molecules in animals, like neurotransmitters, growth factors, and some hormones, induce responses by increasing the concentration of calcium ions in the cytoplasm, which is actually more common than the cyclic AMP pathway we just looked at. The increased calcium concentration can lead to muscle cell contraction, secretion of molecules, and cell division. Cells do contain low concentration of calcium ions because calcium is actively transported out of the cell and actively imported from the cytosol and into the ER. So therefore, the really small changes in concentration can make really huge differences, and that this calcium is usually going to be released from the ER, not pumped into the cell from the outside, which we can see in this diagram here. We have lots of calcium ions on the outside, relatively low on the inside, but we do have higher concentrations of calcium both in the ER and in mitochondria. And so those will be pumped into the cytosol from those organelles, and then that slight increase in concentration will give us a response like maybe a muscle contraction. Pathways that lead to calcium release involve other second messengers like IP3 and DAG. They both are produced by a cleavage of a special phospholipid in the plasma membrane. So here is yet another example of calcium and IP3 in signaling pathways, which you can review on your own. It's pretty similar to what we've seen since the beginning of this. So I hope you are noticing that theme of like this happens and then, a couple of different things happen to activate other things, which ultimately lead to the response, which we are now at. Response is the final step in the signaling pathway. It ultimately leads to the regulation of one or more of the cell's activities. The final response can occur in the nucleus, affecting transcription, or in the cytoplasm, affecting literally anything else. First, we'll look at responses in the nucleus. Many pathways regulate protein synthesis, meaning they will turn a gene on so that it can be transcribed and translated into a protein, or turning a gene off, stopping that process completely. An example would be an activated steroid receptor. The final activated molecule acts as a transcription factor. Transcription factor meaning it is encouraging transcription to happen, so we will have that gene made into a protein. Transcription factors will either turn a gene off or turn a gene on, and we'll look more at that regulation in Unit 6 after winter break. Next, let's look at some cytoplasmic responses. These will regulate the activity of the proteins that have already been transcribed and translated. So one example would be that the signal leads to the opening or closing of an ion channel that's in the plasma membrane. Another example would be that epinephrine activates an enzyme that breaks down glycogen into glucose so that it can be used. And a final example of a cytoplasmic response would be time to divide in cell division, which is what we're looking at next after this. Not in this video, in a separate video. That would be too much. The extent and specificity of the response is regulated in many ways. So one is by the degree of amplification that a signal depends on the function of the specific molecules in the pathway. The steps in a multi-step pathway do provide some control points. The overall efficiency of the response is enhanced by the presence of scaffolding proteins, and terminating the signal will all help regulate the response, which you can see pictures there. Apoptosis integrates multiple cell signaling pathways, and we're just going to talk about this very briefly. Cells that are infected, damaged, or that have reached the end of their functional life undergo programmed cell death, which is called apoptosis. The process of apoptosis is as follows. The DNA is chopped up, organelles are fragmented, cell shrinks and becomes lobed. The cell's parts are packaged into vesicles, and then those vesicles are engulfed and digested by specialized cells. And this is really important in protecting neighboring cells from damage. So if you have one that is infected, it goes under this process so then the surrounding cells don't get infected. Although sometimes apoptosis releases those things. Or, if we have one that is out of control with the cell cycle and we have some tumor formation starting, maybe it goes under apoptosis and keeps the other cells from going through that process as well. The signal for apoptosis can come from inside or outside of the cell. If it's coming from outside of the cell, signaling molecules released from other cells initiate the signal transduction pathway that activates the genes and proteins necessary for apoptosis. Inside the cell, if the DNA becomes too damaged, a series of protein-protein interactions can pass a signal on that triggers a response, which you can see in section 11.5 for examples in soil worms, C. elegans, and other animals. But I don't want to spend time on that right now. You can do that on your own.